welcome to tonight's live stream. I'm delighted to be with Dr. Paul Nelson again, and we are here back with part 3 of orphan genes and other taxonomically restricted genes. So, Paul, this has been a fabulous, series. I was actually talking to this academic maybe about an hour ago just about this series, and the feedback I've actually got from people with a background in philosophy and background in science, this is really unique, right? This is not the sort of stuff you're going to get in your standard textbook.
Well, good. I'm glad that I've got something to offer that's distinctive and worthwhile, and I hope this next part will be the same. I'm going to try to finish up in about 45 to 50 minutes, leaving time for discussion. And I guess we should just jump right in.
All right, let's do it.
So here's my outline. For parts, the first is a concept is missing from the basic categories of comparative biology, which I find really interesting. And I'll present in a few moments a matrix, a theoretical matrix with 4 quadrants that will show you where that missing concept is and what the consequences are for not having it around. Then we're going to talk about something I'll call primary differences and their relation to the principle of continuity. Last year in some talks I gave on this channel about testing universal common descent, I discussed the principle of continuity. I want to reintroduce it here because it's relevant to the topics that we are looking at about gene homology, orphans, and so forth. Then I want to begin to talk about BLAST, which is today, I would say, still the most widely used comparative tool to assess gene and protein homology. I'll go today into the first part of my presentation on BLAST. Next time, part 4, we will look at the critical parameter known as the expect value or the E value. But rather than give you the full tutorial on BLAST today, which frankly would be a little bit tedious, we'll break it up into 2 pieces. We'll look at the beginning of the BLAST tutorial today, and then next time we'll look at the second half. And lastly, because I love thought experiments, we'll end with a mini thought experiment to provoke your thinking for the next session, so that I'll have you coming back next time. So the core principle for today, and it really remains the core principle, I think, for the whole series, is this. Homology is a biologically meaningful concept only if we can also say when and what entities are not homologous with each other. In other words, in order for this concept to have testable content and not to swallow up the whole of comparative biology, we need to be able to distinguish homology from what it is not. And you can see that when we look at this problem of the missing concept. So I want to go back to an article published 23 years ago by W. Doolittle, who is a molecular evolutionist, quite prominent figure at Dalhousie University in Nova Scotia, and actually is a foreign member of the U.S. National Academy of Sciences, someone from whom I've learned a great deal over the years, actually going back to my graduate school days. I look forward, I hope, to meeting him someday. But in this paper, he's talking about homology, which he said is a funny word. He says, in the context of proteins and genes, it makes sense that it's homology only if we don't think about it too deeply. If our model of evolution invokes a single cell as the universal ancestor, then we might conveniently trace all modern genes back to one or another particular family founder gene in the universal ancestral genome. And I think that's probably, I'll say parenthetically, that's probably the case for most biologists who don't, as Doolittle intimates here, who don't think too deeply about homology. They see genes and proteins having lineages working their way back to the set present in the last universal common ancestor. And Doolittle goes on, he said, the universal ancestor seems to represent a sort of horizon beyond which we can justify not looking. Then he goes on the very next part of the article to say something that really should shock you or wake you up. Let me put it that way. He says, if there was no ancestor, no last universal common ancestor, that is, how can we avoid thinking about the possibility that all genes are ultimately derived from a single short RNA, the first replicating ribozyme? If this is true, all genes are homologous. Now that emphasis there in the yellow box, that's in the original. That is for Doolittle sort of knocking on your window saying, hey, you need to consider the possibility that all genes are homologous. Now, I'm willing to entertain that. As a student of comparative biology, I'm willing to entertain that possibility, but frankly, I think it creates grave problems for evolutionary theory, or more generally for biology. Because what happens is if you adopt Doolittle's proposal here, you lose the ability to discriminate phenomena. You can no longer say genes X and Y or genes X, Y, and Z are homologous and genes P and Q are not. One wants to be able to make those discriminations for all kinds of reasons. And I think this proposal that Doolittle is floating here raises grave difficulties. You can think of it on the metaphor of a precipice. You're walking along a hillside and suddenly you come to a cliff and you need to be very careful not to step over that precipice. So let me give you some examples from other bodies of knowledge where we have well-defined distinctions that do work for us. So for instance, consider the whole numbers, the natural numbers in so-called counting numbers in mathematics. We distinguish odd and even numbers via their divisibility by 2, right? Divisible by 2, get 2 equal numbers, not divisible by 2 into 2 equal numbers, even and odd. Or in chemistry, we distinguish elements as we see them, for instance, in the periodic table, from compounds. And these definitions, these concepts and their associated definitions do important work for us. We need for them to be stable, to have a coherent science of chemistry. Well, in biology, we need stable definitions that are coherent with respect to some larger theory. Again, because we want those concepts to do work for us in a way that we can count on them being there the next day and not changing their referent, not changing what they talk about in the natural world. So classically, homology, since Darwin is defined as sharing common ancestry, entities, whether genes, proteins, anatomical structures, right on up to behavior, we consider those things homologous if they trace back to a shared material ancestor. Then the contrast class in this case would be homoplasy. Entities are not homologous if they do not share a common material ancestor. So my point here philosophically is to stress that that giving up stable definitions for concepts like homology comes at a cost, and it's a cost one needs to be very certain that you're willing to pay. All right, now I've shown this cartoon before, and I put it up here to say that this sort of at a first approach does a lot of work for evolutionary biologists. We have homoplasy on the left there, homology on the right. And at this level, these concepts seem stable, and again, sort of thing that one doesn't have to worry about. But let's now apply Doolittle's own thinking to that cartoon that we saw just a moment ago. So this is a figure, and I've shown it before, from one of Doolittle's articles published over 20 years ago now in Scientific American. I don't think he drew this. I think this was actually some staff artist at Scientific American. But what I want you to notice here is the rooting of this wild kind of mangrove-like geometry for the early history of life. And notice that these lineages are not converging to Luca. They are not coming together at the last universal common ancestor. What he has here in the figure, Doolittle describes as a common community of primitive cells. Now, this, I believe, and I think I'm right in saying this, is Doolittle's own drawing. Across the top there, you see the three major domains, and you've got this wild interfingering of lineages with, for instance, bacteria crossing over into eukarya to provide in the endosymbiotic, according to the endosymbiotic hypothesis, the progenitor for the mitochondria or the progenitor for the chloroplast. Again, what I want you to notice is the base of the figure. At the base of the figure, you have at least 5 independent, there may be more, it's hard to tell in the drawing, but I'll assume 5 independent starting points. Independent, that is, in the sense of arising spatiotemporally from separate theaters or environments on the early Earth. In the prebiotic early Earth, these events are happening in discrete or separate settings, meaning that these blue arrows here represent causal pathways that are not in communication with each other. Now, this raises the question, did these independent progenitors give rise to the same molecular structures? For instance, let's assume that these are alkaline submarine vents, a very popular hypothesis right now for the origin of the first biomolecules. That is, that's the environmental setting where those events occurred. In those independent settings, these alkaline submarine vents, are ribosomes being produced? Are nucleic acids like DNA and RNA being produced? Are proteins being produced? If the answer is yes, then the definition of homology as due to material descent is false, because in this early Earth setting, there is no material descent as we understand it today, of one yeast cell giving rise to another, one E. coli splitting into 2, and so forth. That biological descent causality relation does not exist on the early Earth, yet homology, if we answer this question yes, homologous structures are being produced by prebiotic chemical pathways, meaning, to carry it through, the definition of homology as being caused only by material descent in a biological sense is not true. Or we could say no. Those independent settings are not giving rise to the same molecular structures, which makes it very hard indeed to see how they could possibly successfully interact as the story of the origin of the 3 domains goes forward. How could there be viable horizontal gene transfer, for instance, if the ribosome being produced, let's say, in submarine vent 5 is fundamentally incompatible as a molecular machine with the ribosome being produced at submarine vent 4 or 3 and so forth. So you can see, and I don't have this slide in this presentation, but in previous presentations, I've said that the conflicting demands of the naturalistic program of explanation for things like homology with respect to the origin of life and everything that's involved can create deep contradictions in the science of evolutionary theory. Now, one of the good things that Luca does for you as an entity is it kind of cleans the table, cleans the tabletop. And even if you have, as this cartoon illustrates, multiple independent pathways arising in the prebiotic milieu, at Luca, you've got a bottleneck where all lineages but one are swept off the scene. Okay. Now, what happens if, as for Doolittle proposes, we eliminate Luca as a bottleneck or as a theoretical entity that's going to tie together all observed organismal diversity post-Luca? So we'll get rid of Luca. Now you have the possibility of multiple independent lineages arising. And in fact, even though this is a minority viewpoint within evolutionary theory, or it was, that is, in most of the 20th century, it's less so. There are more people holding this view today than when I was a student, and certainly more people holding this view today than during the hegemony of neo-Darwinism in the middle decades of the 20th century. Figures like Stuart Kaufman or Christian de Duve, and I won't list all the theoreticians who have this view. Multiple independent lineages are a live possibility if you raise the probability of the origin of life, moving it away from zero. Sort of the Jacques Monod view, we're just lucky to be here. Move that probability away from zero and over towards one, where it's deterministic or quasi-deterministic, more likely than not to happen. Multiple independent lineages become a live possibility. And under that circumstance, one's definition of homology has got to change, because there is no shared material descent as these lineages cross the threshold from the prebiotic state to the living state. Okay, what's important about this figure that we need to worry about? The concept of homoplasy refers phenomenologically to observed similarity. We say two features, they don't have to be gene sequences or proteins, they could be anatomical characters, exhibit homoplasy because they are similar, but they do not share common ancestry. Now we're coming to this critical problem of a missing concept. So homoplasy really is used in systematics, in comparative genetics, and so forth with respect to misleading similarity. Similarity that is not giving you true history. Okay, so here's a matrix. I mentioned this at the outset that I think is very helpful. We have common descent and no common descent across the top there, and then we have similarity and difference as observed properties of some living system, which gives us a 4-quadrant matrix. Now, the familiar parts of the quadrant are these: common descent causing similarity. All right, that's in fact that causal aspect has now post-Darwin been incorporated into the definition of homology. So the contrast, of course, as I showed a moment ago with the other cartoon, is homoplasy, or using a perhaps more familiar term, an earlier term, convergence. These categories are familiar. So you have similarity caused by common descent, or similarity where there is no common descent, but perhaps natural selection has driven a morphology, let's say, towards a particular form. So in terms of their overall shape, the fusiform, streamlined shape that we see in dolphins with a dorsal fin and so forth, pectoral fins, we also see in sharks, right? They, to the terror of many swimmers, from a distance, a dolphin fin cutting through the water can look very much like a shark fin cutting through the water. And you want to, you need to worry about one and not worry about the other. But you see the convergence in form because that particular morphology is adaptive, functionally useful in an aquatic setting. All right. This is textbook orthodoxy, that upper left quadrant. Common ancestry from Luca is the most parsimonious explanation for the universally conserved similarities that we see in organisms. And over here, Darwin knew all about this. In The Origin and other of his writings, he talks about adaptation to similar circumstances environmentally, giving rise to similar structures without material descent. In that case, the signal of common ancestry is diminished, and you face the problem of sifting homoplasy from homology when you want to build your trees of life. Because one of them is giving you real history, namely homology, and the other is giving you misleading history. Now, down here, we have divergence. And again, this was a key part of Darwin's case, that in the relation of descent from parent to offspring, that cycle of generation, modifications occur. As those modifications arise, let's say from the common ancestor of all mammals, some, let's say, small insectivore managed not to get stepped on by a dinosaur, and you have the KT impact, the dinosaurs lose out and the mammals flourish. And the basic form of the mammalian common ancestor then radiates during the tertiary period into, whales and bats, ungulates, the whole amazing range of mammalian orders. There are shared characters, of course, that define mammalia, but you have tremendous divergence anatomically within that clade. Differences. Now, these concepts should be familiar to you. They fill your textbooks. We have a quadrant that is open. This one. Why? What's in there? Now, this is a difficult question to answer, I think, for many evolutionary biologists. It was a difficult question to answer for me as a student of evolution. This concept appears to be missing from evolutionary theory. And that, let's give it a name. We'll call it primary or original differences, where you do not have common descent and you have differences that are present at the start. They do not arise via divergence from common ancestry, because there is no common ancestry at this point. You might say, well, evolution doesn't need a concept like that. That's why it's missing from your little matrix here, Paul. Actually, it probably does need this concept. And the fact that it's missing, it's not present in textbooks. I'll encourage you to go look at a good evolution textbook and see if you can find something about primary or original differences. My guess is you will not. Thoughtful theoreticians, such as Ford Doolittle and John Maynard Smith, the late John Maynard Smith, were well aware that primary differences may be biologically necessary. So we've already seen this quote from Doolittle. What he's describing in this passage from his paper in 2000 is, as you work your way back towards the last universal common ancestor, you seem to need a certain irreducible amount of molecular hardware, what he calls these protein superfamilies. And in an earlier talk, in the talk, I think the one preceding this one, available at YouTube, I described this as the homology horizon, where and I'll say more about this in just a moment, where the folds, the protein folds that give you precise enzymatic reactions, for instance, polymerases, that will give you, that will transcribe RNA from DNA. so an RNA polymerase or a DNA polymerase. Compare that to the proteasome, the cell's garbage disposal, or the enormous molecular machine, the ribosome, to me, the molecular machine defining the living state on planet Earth and so forth. These molecular tools are not the same. And when you go to the protein data bank and look them up, it's crystal clear these are as different as a hammer is from a saw, from a drill, and so forth. And what Doolittle says in this passage, which I find I've gone back to it multiple times during these presentations because it's so full of insight, is you seem to come to a horizon where you can't go beyond that and describe a functional cell. Much earlier, in fact, in 1970, John Maynard Smith saw this. He said, we come to a horizon, I'm paraphrasing, obviously. We come to a horizon where we seem to bump up against a set of starting materials, if you will, or to be more precise, molecular actors, molecular players that we have to presuppose, and we cannot go beyond them. And it's very difficult to see how that starting set arose, particularly if you examine what's necessary, as we now know in 2023 via things like transposon mutagenesis, what's now necessary for the living state. So to summarize this, the functional demand for primary differences in the first cells seems to require a theoretical grounding distinct from the customary moves in evolutionary reasoning, which, as I said in an earlier presentation, are copy and vary. You can think of these as macros or command sets. Make copies of yourself, vary those copies. That is natural selection, or frankly, that's any evolutionary process. You need to be able to make copies, and those copies have got to be able to vary. So how do we know this? How do we know that the functional demands of the living state require primary differences? Well, we know it from experimental and observational biology, from the demands of the organismal state itself. So I showed this cartoon before. I wanted to bring it here again. Here are 4 proteins demonstrably necessary for a bacterial cell. You've got a chaperone complex there in the right, a cell division protein, an elongation factor, and I forget what the 4th one is. I apologize for that. But these are only a small fraction of the set of any known bacterial cell. And this has been run pretty thoroughly to the ground by Craig Venter and colleagues in their SYN 3.0 cellular engineering that they've done. And I think in the field of synthetic biology, it is a standing puzzle, research puzzle, how stripped down can the set of components of a viable cell be such that you can keep it alive in culture? Well, you bump up against a pretty substantial set of diverse molecular components. And again, I encourage you, take the parts list, so to speak, of mycoplasma and go to the protein data bank and just download a dozen or so, just randomly pick proteins and download them and look at the 3-dimensional structures. And what you are going to see is a great deal of molecular diversity. So let's tie this now into what I call the principle of continuity, because it's very closely connected to how we're going to understand homology and the demands that biology makes on our understanding of homology and its contrast classes, homoplasy and what I'm calling divergence. So if you go to to the YouTube page that Subor maintains, in August last year, I talked about this in more detail than I'll be able to do today. But because the concept is related, this notion of continuity is related to what I'm saying today, I wanted to revisit it. So how do primary differences, as I've just sketched them, relate to this notion of the principle of continuity? I'll give the view as it connects to universal common descent, because that's another aspect of what we're talking about today. Universal common descent is just about as abstract a notion as you can find in biology, for all kinds of reasons that I'm actually writing about right now. It gives you a formal geometry of relationships that has Luca at its base. All right. The last universal common ancestor is the ancestor present in the set of ancestors of any living thing on the planet. Very elegant notion in that respect, right? It's our ultimate grandmother, if you will. And it's the only member present in all such sets. Now, this formal geometry, it may surprise you to hear this, doesn't actually predict anything until you couple it to everything else we know in biology. So we'll couple universal common descent to what I'm calling AUX. By that, I mean an auxiliary theory. How do we get testable consequences from this formal geometry? We couple it to the rest of our biological knowledge, and that lets us make predictions. And this is a feature of universal common descent that's not widely appreciated, but I think if you look at textbook predictions from universal common descent and pull them apart logically, you will find that the prediction flows from coupling that universal geometry. It's a formal, mathematically, it's a Steiner tree with a single root. Couple that to everything else you know in biology, and that will yield predictions. And you can, because of the universality of the geometry, the predictions then can go right across all domains of life. All right. Metabolism is necessary for cell survival. There's a strong prediction. Or let me put it this way. There's a strong piece of knowledge that comes from, let's say, cell biology or organismal biology. It doesn't rest on evolutionary theory. We learned it really more or less independently of evolutionary theory. All cells require ribosomes. We learned that from cell biology and from the details of molecular genetics and so forth, right? These machines need to be able to take nucleic acid, turn it into protein. How are you going to run your metabolism? Well, you're going to need some kind of proton gradient, right? That's going to be essential to maintain you out of equilibrium with your environment, to do work and so forth. These pieces of knowledge, if you unpack them, you will find they're not derived from evolutionary theory. I remember I got to know Will Provine before he died. It was a wonderful guy and very outspoken, remarkably so. And he gave a talk when I was a student at the Field Museum Spring Systematic Symposium in the 1980s, in the late 1980s. And those who know Provine know that he loved to provoke. He was a provocateur, right? A gadfly. And in this particular talk, which I'll never forget, I was sitting right down in the front of the auditorium there at the Field Museum here in Chicago. He said, you can be a world-class biologist and basically know evolutionary theory. You just don't need it. Now, he was intending to scandalize his audience, and he did. I mean, the auditorium was packed, and I didn't hear anyone boo, but I heard a lot of sharp intakes of breath, like, what on earth is he saying? The point he was making is that much of biological research, much of biological investigation can get well down the road, largely independent of evolutionary theory. Now, evolutionary biologists complain about this regularly. And I think with some justification. I think there's a lot of evolutionary theory that in fact, depending on the field you're working in, if you're a virologist, you better know some evolutionary theory. If you're an immunologist or someone on the medical side in infectious disease, evolutionary theory is an essential tool, right? Provine's point was the connection between evolution and large areas of biological research is rather tenuous. Okay, so anyway, I'm spending too much time on that point. The second point here, B, is the one that really matters. If you think about metabolism or the structure of the ribosome or what goes into a cell wall, even in a bacterial cell, if you think about the parts, the actual molecular parts that are required, they're diverse. They are, and there are primary differences exhibited by them. Now, you might say, well, Paul, that's a modern cell. That's the product of almost 4 billion or 3.8 billion years of evolution. Okay. Describe for me a cell without that stuff, without that molecular diversity. What you will find is your description is going to be evanescent, a will-o'-the-wisp. There will be relatively little actual content because the cells that we know, I'll come back to this point in a few slides, the cells that we actually know exhibit tremendous molecular diversity. All right, this brings us to the principle of continuity. And you can see, I hope, how primary molecular differences interfinger with the principle of continuity, because there are some transformations that are possible in biology and there are others that are not. And this is one of the ways that actually evolutionary theory ends up being tested. So the principle of continuity is simplicity itself. It says, if you hypothesize a transformation from A to B, so that could be transformation of a protein fold, that could be transformation of a gene sequence, let's say with a protein product, or maybe it's a regulatory element, some pathway of transformation where A and B are sufficiently distinct from each other that you'll give them separate names. If that's your hypothesis for how B came to be, where A is the ancestral form and B is the descendant, then every point in that pathway must be biologically possible. Now, this may seem to be a truism. In fact, it's very powerful. When you go into the evolutionary biology literature with the principle of continuity in your consciousness, you will find it present everywhere because it is a key tool, to give it a name, that evolutionary biologists use to judge the merits of each other's hypotheses. So Leslie Orgel, in a famous paper in 1968, I think published right next to another famous paper by Francis Crick. I believe at the time Orgel was at the Salk Institute. And he's looking at the question of the origin of the nucleic acid protein system with which we are familiar at the origin of life. Orgel was an RNA chemist, biochemist who worked on the origin of life. And in this paper, he said, how am I going to sort out all the hypotheses that confront me? I'm paraphrasing, obviously. How am I going to sort out all these hypotheses with respect to their relative plausibility? Well, he says, I'm going to be guided by the principle of continuity. Why? Because evolution has to be continuous. There's no dying in evolution. If you go extinct, it's over, right? So if you're going to be along one of these pathways of transformation as a viable organism, you've got to be able to do so, leaving offspring, maintaining continuity. This principle allowed him to pass judgment critically on a hypothesis current at the time that the original biochemistry, if you will, capable of replication, could have undergone a fundamental chemical transformation to the system that we know today, DNA, RNA, protein. And he said the problem is, along that pathway of transformation, there will come a point when all the information you accumulate in the unknown previous biochemistry that is not based on nucleic acid, 20 amino acids, and so forth, all that information will have to be transferred from one kind of chemistry to another, keeping yourself alive, as you go. And he said, I just don't see any way that that's possible, because you're going to cross a gulf of biological impossibility where in a sense, you're neither fish nor fowl. All right. Orgel applies this with respect to the origin of life and the origin of terrestrial biochemistry, the biochemistry that we know on this planet. But you can find it everywhere in evolutionary theory, from the origin of life right on up to the origin of language and Homo sapiens. Principle of continuity is a powerful tool. And if you take it seriously, as I think you should, because it's really everything else we know in biology, it becomes the ultimate referee of universal common descent truth. Okay. Now, how does this come home or work its way back around to where we were talking about molecular similarity? Well, Massimo Di Giulio is an evolutionary theorist in Italy who, over the past, I would say, decade and a half, has been arguing that Luca probably did not exist. And his reasons for that have to do with the principle of continuity. Namely, what kinds of transformations are possible in cell biology. So in this paper in the Journal of Theoretical Biology 5 years ago, he said, if you began with what he calls a genote, I'm not quite sure how to pronounce the word. That is a cell that has more or less the properties that we associate with cells today. The contrast class to a genote or a genote is a progenote. A progenote, I would argue, and this is, I guess, a critique of Di Giulio. I don't want to press the point, but a progenote has actually never been described, to my knowledge. I think the concept originated with Carl Woese, but I have never actually seen a description of a functional progenote. It is a hypothetical entity to have some replicating system prior to life as we know it that can undergo evolutionary processes like selection. And Di Giulio says a transformation of a fundamental cellular type, let's say an archaean to a bacterium, or either of those to a eukaryote, he says, seems impossible to accomplish. Why? Because you would have to change fundamental traits that are tied to the living state for that particular cell type. He says this cannot have been realized. So you can see what he's doing. He's applying a principle of continuity to this early phase in life's history. And here is a figure from his paper where you have the progenotic era, progenotic Luca. It's not an actual cell. It's a community of cells. Again, it is a hypothetical entity, or these are hypothetical entities. They are never actually described in any way that would be functional that I know in his work, but they're placeholders for a theory. Notice here that these domains, bacteria and archaea, are arising independently of each other on this surface. So time is going this way. Here's an easier way to look at it. Time is going this way from past to present. And these groups do not share common ancestry in any sense that we can understand, right? In any sense that you would observe in a chemostat with E. coli dividing, as they do, right? You give them nutrients and they're going to make more E. coli, right? These groups are arising independently of each other. And the important thing to realize are these are the cells that we actually know. Go past that threshold and you go into a realm of hypothetical entities. Now, why does this matter? It matters because if you're going to describe these points, which are arising independently of each other, these common ancestors for, in this case, I think that, as I recall his hypothesis, within the bacteria, he has multiple independent origins, and in the archaea. If you're going to describe them, you have to find a way to relate their properties one to another, which means you're going to need criteria of homology that make sense of the fact that these groups are arising independently of each other, which brings us to BLAST. And I think I will finish on time if I go really fast. I apologize for that, but I do want to try to finish on time. So I've called homology determination using BLAST a bioinformatics convention. Now, what do I mean by that term convention? Well, a convention in science is a definition, method, or practice that is employed for a variety of reasons. And many of them are not good. I'll be honest, these are not good reasons, such as habit. Everyone who goes into science, in my case, the science that I studied most intimately was systematics and evolutionary theory. You pick up practices, disciplinary practices from your environment, from your teachers, from your fellow students, from your textbooks, from the literature, from everything that you soak up. You pick up a set of tools, and some of them are good and others may not be so good. It's hard to sort that out when you're a student, right? You're just learning. This is disciplinary inertia. And as I said, some of those tools may be useful. Some of them may be reliable. Others may be crap, to be blunt about it, right? My point is, disciplinary inertia is something that you find yourself in. You're in a stream of thinking. As I don't want to quote from German, so let me stick with good old English. It's a set of practices that you learn and adopt more or less because you can't help yourself. All right. Well, related to that is this: conventions are easy to use because everybody else is using them. This is clear when we leave the realm of academics and go into sports, right? There are all kinds of conventions in sports. And, if you're a fan of baseball, you're a fan of football, ice hockey as I am, and so forth, you know that there are conventions in how things are done, all right? And some conventions are easy to use, and so people use them. Now, this is the worst aspect of a convention. Conventions are often used without any underlying theoretical rationale. And you can see again how these 3 bullet points relate to each other. If you use a convention because everyone around you does, and no one has ever bothered to justify it, it's just the way that things are done, you may find yourself waking up one morning and saying, why am I defining homology like that? Or why am I using a cladistic method that presupposes this? And you say, I really need some rationale for that is independent of the fact that I just happened to do it, that I learned it. It was my, the environment intellectually in which I found myself. When you are using a tool in science without an underlying rationale or justification, that's very dangerous. Now, conventions can survive for a long time, as long as nature, if I can personify her, as long as the evidence corroborates. Often, as we see now, today, in genomics, comparative genomics, and evolutionary theory, there's an ocean of data that nobody expected, and many of the conventions, in particular the ones that animate BLAST or that give BLAST its form as a tool or practice, those conventions are bumping up against anomalous data. And if you continue to use the convention in the face of anomalous data, that can engender theoretical incoherence, and your science can end up in big trouble. All right, so that's what I mean by a convention. All right, let's look at BLAST. We're almost done. Then we'll conclude with my little thought experiment. Basic local alignment search tool. Now, this paper is one of the most widely cited in my study of the field, right? I just checked Google yesterday, Google Scholar. over 100,000 citations. And it's a case of Altschul and colleagues building a tool that people needed, and scientists are intensely pragmatic. They migrate to tools that are useful and that other people are going to use as well. Very few, in my experience, very few biologists who use BLAST ever bother to look under the hood. So there's an analogy to an automotive mechanic, right? You pop the hood and look under, and it's a V8 or some different kind of engine. I bought a Honda Pilot. My wife and I bought a Honda Pilot a few years ago. I love this car, and we were at the dealership, Honda dealership here in Northern Illinois, and I went to open the hood just because I feel like, well-informed consumer, I should look at the engine, at least look at it, right? I go to open the hood, and the salesman goes, "There's nothing for you under there." That's all our business. The only reason you want to go under there is to replace the wiper fluid. I felt put in my place because, in fact, what do I know about engines? Nothing, right? So in this case, however, we should look under the hood with this method and not simply use it. Now, very few people know this. I didn't know this until I went and looked. I got the original BLAST paper, 1990. When BLAST was formulated originally, it had relatively little to do with evolutionary theory. It was a tool for getting function clues from sequences, right? So in 1990, this is well before, 5 or 6 years before automated DNA sequencing, but data are starting to accumulate. Protein and sequence, DNA, nucleic acid sequence data are starting to accumulate. And you want to have labor-saving methods of finding out what your sequence might be doing. Well, you go to the database, the databases that were beginning to grow quite rapidly in the 90s, and you look for clues. What do I have here? Is it a kinase? Is it a structural protein? Is it a regulatory element? How do you do that? It's just the thing that you would do with this dictionary, right? I'm chugging along through some text. I find a character string that I've never seen before in English. I actually have the Oxford English Dictionary up there, the compact edition up there on the shelf for the really hard ones. But this, my Random House desk dictionary, is a pretty good representation of English words. And you could think of this as a database of functional modules in natural language. Well, the same holds true for protein and nucleic acid databases. Why does function matter as an empirical target? Well, it matters because we can see it. We can observe it directly. Lee Van Valen, in a publication that I've now cited a few times in these lectures, says, and he's right, homology, evolutionary history, we do not observe, or only rarely, only under very rare circumstances, such as we've had over the past 3 years with SARS-CoV-2. A real-time experiment in virology and molecular evolution for an RNA virus, right? We can observe in real time how that sequence is changing around the globe. In most cases, however, homology is going to be an inference, not an observation. Function, you can, in a wet lab, you can figure out what your protein is doing, okay? So here's your database. In a little cartoon, there's my new sequence, and I look for matches. Just the way I would if I went to an English dictionary. Ribosomal protein, now I know what my new sequence is doing. And of course, this can be automated. And it is. Maybe next time I'll talk about some of the problems that arise. Kara Weissman in her work has pointed out some of the problems that arise in using different automated methods of annotation for sequence data, where you get different answers depending on the annotation program that you're using, but I won't let that slow us down. Now, This is important. This is an important slide. There can arise fundamental conflicts between the differing demands of your inquiry with respect to homology. So when BLAST was originally formulated, it was, as I just pointed out right there in the opening of the paper, the original 1990 paper, they were looking for functional clues. Align sequences, try to gather information about what your sequence is doing. You can only do that at a relatively shallow or very, very shallow depth, however, with respect to evolution. So you can project those functions, let's say these are two genes, A and B, backwards in time, but you will find that you have conflicts with what evolutionary history may be demanding of you. So I showed this slide last time, but I couldn't show it in the sequential way that I'm doing today, so I'm doing it again. As you go back in time, the number of entities to which you can appeal to build your evolutionary trees goes down rapidly. They just haven't evolved yet. And you may find that your functional descriptions and your evolutionary descriptions are in conflict with each other. So these red circles here indicate points of conflict where my functional hypothesis is going to crash into my evolutionary hypothesis. Because in general, evolutionary scenario construction says as you go back in time, infer smaller, simpler, fewer entities. They haven't evolved yet. And the functional description may find itself in conflict with the evolutionary description. In fact, again, I don't have time to go into this today, but that is a real problem. Now, let's go back to the BLAST algorithm, and I'm almost done. I think I might run over by 5 minutes. I apologize for that. BLAST comes in many flavors. Probably the most widely used is BLAST-P, which is sequence comparison using amino acid data. P for protein. BLAST-N is nucleotide data, and then there are a variety of BLAST tools that give you multiple reading frames. These are not, to my knowledge, as widely used, but the point is, BLAST has lots of different versions since it was originally formulated. For simplicity's sake, for today, we'll look at BLAST-P, which is the original form of the algorithm. So here's the basic protocol. And of course, this is done via computation. The investigator typically will not be doing this. He or she will be inputting sequences, and the algorithm is going to do these 6 steps. So you'll input your query sequence. Want to know what it is? The algorithm will break the query sequence into words to give them a name. It will look for synonyms, or it will generate, synonyms of those words using substitution matrices. I'll explain what those are in a moment. Scan the database for hits to these words. Extend the hits in both directions. I'll go through this in just a moment. And then give you the best matches, rank ordered. Okay, so here's the standard format for an amino acid sequence. Each of these is a single letter representation of an amino acid. So this is a ribosomal protein. Here's a nucleotide sequence, and this is FASTA format, so it's a standard format. So here we have the name of the protein. Here's the name of the gene. This then is a DNA sequence, and this is a standard format that's machine readable. I'm just going to skip through this slide. Now, the query sequence is broken up into words, shorter modules. So in an amino acid sequence, those are every possible 3-letter combination of amino acids. So you can see how these overlap as you cut them up. And that is what the algorithm will then use as it goes into the database. And this is a protein sequence, obviously. These amino acid words will be the bait, if you will, that will be submitted to the database looking for hits. Now, because proteins vary and amino acids can substitute for each other in protein assembly and protein evolution, you need to have synonyms for the words to make sure you're casting your net. I apologize for mixing metaphors. To make sure you're casting your net widely enough to get relevant hits. So there are two substitution matrices that have been used with BLAST, Blossom 62 and PAM. These are theoretical, I guess you could say, tools of manipulation that allow you to expand your search where, let's say, some big amino acid with a big ring or something small like glycine can have other amino acids substitute for it in a protein structure without disrupting that structure too very much. And there are in the substitution matrices, there are penalties assigned depending on which amino acid is being substituted. In any case, this allows you to expand your search space and catch more homologs. So I'm going to skip that slide. Now, these matrices are based on evolutionary principles. And next time, I hope to go into this in a little more detail. But all I wanted to indicate today is a significant amount of evolutionary theory is built into BLAST, sort of baked into the algorithm. Whether you find that objectionable or not, I guess it's a matter of taste. For me, it's something that I worry about, frankly. And next time I'll say more about that. Anyway, the algorithm scans the databases for hits to the words. When it finds them, it begins to extend the search in both directions, 5' and 3' along the sequence, and it keeps a score. And the score has to go in the right direction to extend the search. If the score begins to drop, the algorithm, in effect, says, I'm not getting homology here. Skip this sequence, go to another one. All right. So if the alignment does not exceed a preset threshold of similarity as you're extending the hits along the sequence, it's going to be dropped because these searches are computationally very expensive. All right. That's sort of the first bit of what BLAST does. Next time, we'll talk about how these searches are scored and how all of this factors into what's known as the expect value. All right. That's for next time. My mini thought experiment.
Brilliant.
All right.
That was amazing. Especially the BLAST stuff. I did not expect that. That is I think some really novel research. It just got me thinking. I mean, earlier you mentioned Will Provine, and I was thinking of the Johnson debate of spring '94. And, it's not, I mean, these conventions that you referred to earlier and how they're baked in, it is really, I mean, you're not going to look under the hood if you subscribe to naturalistic philosophy, because you don't want anything, you don't want to find anything, essentially. I mean, that's the way I would put it.
Well, I think looking under the hood is scary because you realize, certainly I did, when I began to investigate BLAST, just how much theory is built in. Theory where I want to say, well, why should I accept that? Why should I accept that substitution matrix?
Yeah.
The substitution matrices actually are the original ones, Blossom 62 and PAM, have been around for a long time.
Yeah.
And in a project that I'm involved in called Orphan Base, we have been troubled, and it's a problem we haven't been able to solve, that we, for a variety of reasons, have had to build BLAST into our pipeline, into our analytical pipeline. And I feel like I would like to be able to take that out. And we actually have a sub-project in our group called Better Than BLAST, where we're trying to find better algorithms, and there are others out there, to do these kinds of sequence comparisons. But next time I will go into much more detail about the theoretical content of what ought to be just, I don't want to use the adjective objective, reliable tools where I don't have to presuppose evolutionary principles to use the tool. I want to discover if evolution worked. I don't want to presuppose that it did.
Yeah.
And then go look at proteins. You know what, Subor, for the sake of time, I'm going to admit the thought experiment because I ran over by about 10 minutes, and it might be fun actually to start the next one with a thought experiment. Brilliant. And there's enough on the table with this presentation, and it's already over an hour. So
no worries.
I'll stop.
No worries. Perfect. So thank you so much for this session. I did actually have a few more questions, but maybe next time we can actually answer them.
Go ahead and ask them. I mean, I said at the beginning I wanted to have time for discussion. So
right.
Unless you're exhausted,
I'm ready to go. I mean, I'm really excited about this BLAST stuff. What's interesting here is, you were earlier speaking about primary or original differences, and that's what's missing from the matrix, right? These types of things you find in the literature. I forgot who you were referring to that actually pointed towards this, but to even think about, all right, what is missing? It's like Sherlock Holmes, right? You had that, you had that famous case where the dog doesn't bark. So he looks for the negative clues. He looks for something that's missing, right? Why do you think a lot of scientists, biologists, even philosophers, they don't look for those negative clues? They're just happy with the conventions the way that they are. They don't want to open up the hood, and they're not thinking in this novel way.
There is a German aphorism that I want to use here.
Go ahead.
And I'll translate it. Wenn wir selbst zu denken meinen, denkt die Zeit in uns. Which, freely translated, means when we think that we're thinking for ourselves, the times think through us. Right? It's very Germanic, right? The idea is that we all exist in an intellectual culture where we're kind of bobbing along. We're being carried like corks in a powerful stream. And it takes great courage and stubbornness and even a little craziness to grab onto something. If I'll use the analogy of a you're being carried down a river, and everybody else is being carried down the river with you, and you decide, I want to stop. I don't want to go that way. And you reach out and you grab a root or a piece of, something, a rock, let's say, and hold on to stop the process of being carried out, down by that stream of water. It takes a lot of strength to do that. And I think that it's all too human to find yourself in a discipline, whether it's philosophy of science or evolutionary biology or any science or academic discipline, and just be carried along by the stream, because it's hard. And one of the things I love about doing these streams, these live streams, and being in contact with people, is there is a community of thought that's developed over the past 20 years where you've got people helping each other, encouraging other people to take a stand and say, I'm not going to just assume that homology has this structure, right? Theoretically, yeah. I'm going to try something different. Doing hard things is hard.
Yeah, it's hard, but it's also, I mean, I was just yesterday listening to Robert Greene's book, The Daily Laws, and he's really into human psychology and this type of thing. And he said something really shocking. I actually took a screenshot because I was like, wow, this is amazing. He said the most cowardly people, in his opinion, are academics, because they will never go against the intellectual heir. They will never they don't want to. I mean, they'll be happy to discover something novel in an area and get an award for it, but the theoretical underpinnings, like you spoke about, the conventions are not even questioned. Nobody wants to do that because it's the hard work, it's the difficult work, and it's actually, you're going to be on your own. When you were speaking, I was remembering a reading on Jerry Fodor's website, Why Evolution is True. His attack on Jerry Fodor when he came up with what Darwin got wrong. I mean, he labeled
Jerry Coyne's attack on Jerry Fodor.
Yeah, sorry, Jerry Coyne's attack on Jerry Fodor, right? And he was labeled on that website a secular creationist, right? And I was like, this is so ideological. Just deal with his argument. And again, Nagel, what happened with him? Jerry said he's no longer going to be welcome on my website. So, I understand why they don't want to do this. And maybe this is also why something like, challenging Blast isn't really going to go down well, but it's going to be needed. And I'm very excited with this stuff.
Yeah, well, as I said, next time we'll finish the story and look at, how you go from the sort of mechanistic part of the algorithm, where there is some evolutionary theory baked in those substitution matrices. The rest of it's just straight up sequence comparison, right? It's just a mathematical operation. The thing that encourages me, and I tend to be a kind of hardcore optimist, is if you can plant in someone's mind I guess it's almost like putting an irritant within the shell of a clam, right? That irritant sits there, that little grain of sand sits there, and in bivalves, they coat it and they produce a pearl. And you have something very beautiful and lovely, a piece of jewelry potentially emerges from an irritant. When I look at Einstein's career and these incredible papers that he wrote in 1905, he was troubled by the understanding of electromagnetism in the late 19th century, and he built a pearl around it. So there are many cases in science where someone just happens to watch a YouTube video from, let's say, Mike Levin at Tufts. And, as I love to listen to his talks because he's such a contrary thinker, right? Where he says, I look at these flatworms that I study, and genetically they're all over the map, yet they consistently produce with very high fidelity the same adult morphology. How is that possible if DNA runs everything? All right, so that idea, graduate student hears the talk, it gets planted as an irritant or a stimulant or something in that kid's brain, and he goes back to his molecular biology lab and he thinks, maybe DNA isn't everything. Yeah, maybe there are higher levels of control. So I'm an optimist for these kinds of swimming against the tide, if you will, or swimming against the current. And it can be done. It can be done.
Sure. I'm a bit worried about your time, so I have two more questions. Should we leave it for next time, or can we do this now?
Why don't we do one more?
One more. All right, brilliant. Thank you for that answer, by the way. That was really enlightening. So one of the things that you said earlier is, primary differences may be biologically necessary. Could you explain that in a bit more detail, please? Sure.
So this comes right out of experimental cell biology. It's a direct result of techniques such as transposon mutagenesis. So in transposon mutagenesis, DNA sequences that have the ability to make copies of themselves are introduced into a cell or a cell population, and they land randomly with respect to, let's say it's a bacterial cell, the circular chromosome of that bacterial cell. These transposons are landing and they're disrupting open reading frames as they do. Now, in some cases, the transposon will land in an essential gene. Let's say the gene is coding for a DNA replication protein or a polymerase or some essential ribosomal protein. You will have those plates will, in your experimental population, those plates will be sterile. There'll be no growth. It's over. Essential hardware was hit. The cell can't make a go of it. It's over. Now, in some cases, the transposon will integrate. It will not hit essential hardware. You'll be able to plate those cells out. You'll have populations. They're growing, right? And you sequence the DNA, and you look to see where the transposons are. The fact that you see a transposon in an otherwise viable cell tells you that at least at that location, under these lab conditions, whatever that transposon disrupted, that gene was not essential. Do that enough, and you can map out, again with a bacterial cell, simple bacterial cell, move around the chromosome where the non-negotiables are, the hardware, molecular hardware that is essential, and make a list of these proteins, go looking for them, and what you will find when you do is they are not the same molecular actors.
Yeah.
They're three-dimensional folds, they're conformations. The functional form of the protein is strikingly different from one protein to the next. Meaning, if you want that bacterial species at all, it's going to require a core set of functionally and structurally diverse parts. Just the way that, well, here's my iPhone, okay? It's got a screen, it's got power buttons, it's got, sort of the chips that are the CPU, there's a little camera and so forth. If I could take, and I'm not going to do it, but go to Apple and have them disassemble this and lay all the parts out on a tabletop, they will not be the same entities. Complex functional systems are characterized by diversity of shape and form when you look at their constituents. So the viewer should think about really any complex system that he or she knows, their car, their bicycle, the refrigerator in their kitchen and so forth.
Yeah.
Go the other direction in your mind. It would be difficult or impossible to do as an experiment, but you can do this as a kind of thought experiment. Try to reduce all the molecular diversity to just a handful of entities that are all the same or that have a high similarity. And what you will find is you will lose the very thing you want. It will no longer be an iPhone. It will no longer be a refrigerator. It will no longer be a cell. So when I say primary differences are functionally necessary, this is simply the deliverance of cell biology to us over the past 50 years. And if you couple that to an evolutionary scenario, you will find very, very interesting problems emerging.
Yeah.
So, for instance, between archaea and bacteria, the DNA replication machinery is not conserved. It's doing the same thing at a high functional level. It's replicating nucleic acid, but the actual molecular players are not homologous. Run that story backwards in time, and this is the reason that W4 Doolittle gave up on Luca and gave up on the single tree picture. He said, you take all this molecular diversity and run it backwards in time, you find that you end up with what he called the genome of Eden. Tens of thousands, hundreds of thousands of different genes have to be present in the last universal common ancestor. And he said that's just not biologically plausible. So I hope that helps. Does that clarify?
That was refreshingly lucid. Thank you so much, Paul. And we'll have you hopefully next week. We'll figure out a time.
Yeah, we'll be in touch. I'm looking at my calendar here. We'll work out something.
All right, thank you for your fabulous presentation as always. Until next time, Assalamu alaikum.